Okay, we're going to start. So we're very pleased today to have Dr. Raghu Mirmira and Dr. Bobbie Jo Webb Robertson joining us to discuss islet stress and its biomarkers in type 1 diabetes. Just a little bio about both of these elite scientists. Dr. Webb Robertson is currently the Division Director of Biological Science at the PNNL in Oregon. Her research is focused on the development of statistical inference models, largely machine learning focused on data integration with application to diverse biomarker discovery problems. Collaboration has been a key component of her career, previously serving as the PNNL Director for the Precision Medicine Innovation Collaboratory, which is a partnership with Oregon Health and Sciences University. She also holds joint appointmentships in the Department of Biomedical Engineering at OHSU, Department of Biostatistics and Informatics at the University of Colorado on Schutz Medical Campus, and the Department of Pathology, Immunology, and Laboratory Medicine at the University of Florida. She has a BA in mathematics from Eastern Oregon University and an MS in statistics and operations and a PhD in decision sciences and engineering systems from Rensselaer Polytech. And our other speaker, Dr. Mirmira, is a professor of medicine, vice chair for research in medicine, and director of the Diabetes Research and Training Center at the University of Chicago. His research is focused on the cellular and molecular pathophysiology of diabetes, particularly the role of the insulin-producing islet B cells. His research work laid the foundation for newly appreciated role of molecular stress pathways in B cells and the induction beta cells and the induction of autoimmunity in type 1 diabetes. His laboratory has been continuously funded by the NIH for nearly 25 years. He's published more than 190 papers. He's well known in the field, and he previously served as the inaugural director of the NIH-funded Diabetes Research Center and the Medical Scientist Training Program at IU. Dr. Mirmira received his MD-PhD from University of Chicago, where he published evidence for rapid-acting nature of monomeric insulin analogs. Thank you so much, both of you, for joining us, and we're really excited to talk about your new sort of research and revelations regarding islet stress and its biomarkers for type 1 diabetes.
Wonderful. Thank you, Monica. I'm Raghu Mirmira, and Bobbie Jo is joining me today, and we want to tell you a little bit about what we're doing. And I should point out, and I was really delighted to see that Wenting Wu from Indiana is also in our audience today, because she has also contributed importantly to this work, and we're gonna recognize her contributions as we go through this. This work is currently being submitted for publication, and so you'll kind of get a preview of what we've been doing. So anyway, I'm just going to provide a little bit of background and context for what we are doing, and then I'm going to kind of let Bobbie Jo take it from there to talk about some of our machine learning components to this project, which has been important. So, so just to provide a little bit of context here, let me see if I can. There we go. I can move forward. Okay, so so this is obviously surprising to nobody, but it's important to always point out that while we talk about the growth of type 2 diabetes in youth today, if you look at youth with diabetes, what you find is that certainly before the age of 10 years, the majority of diabetes, by far and away, 90+%, are still type 1. So, we have the phenomenon of obesity, but just because you're obese and have diabetes doesn't mean you have type 2. And even if you look in the 10 to 19 age group, you'll see that, across the different ethnicities, it's still by and large, type 1 diabetes, although, you start to approach the, the type 2 realm. And where we're at with this disease, diabetes as a whole, is maybe rethinking a little bit how we look at this disease, and that maybe there is a bit more of a sliding scale between type 1 and type 2. There are people that we can identify as type 2 that, have autoantibodies or at least evidence of autoimmunity. And there have been some nice papers recently that have demonstrated the presence, in people with type 2 and no autoantibodies of, autoimmunity or islet reactive T cells in up to 40% of these individuals. So It's just a way of rethinking how we look at the disease overall. And maybe, this sort of binary classification is something we need to reconsider. So when I show you some of the studies that we're doing, I want to be very clear that by and large, most of our studies are done in kids. And beyond that, we still consider this kind of a binary classification, and there are some caveats associated with that. So let's just keep all of that in mind as caveats. So type 1 diabetes, I think many people kind of view it as a disorder in which cells of the immune system penetrate the islet in the pancreas and then start attacking the beta cell. More recently, however, we know that there's actually a dialogue between cells of the pancreas and the immune system. And when I say pancreas, I mean the entire pancreas. What I'm showing here is an islet with highlighting the beta cells, which are really the focus of destruction. But let's think about this for a little bit, is that we now know that there are multiple cell types that contribute to type 1 diabetes. And there are many groups around the country that are looking at not just the contributions of the immune cells, but the contribution of beta cells, even alpha cells. We know that they can be dysfunctional in type 1 diabetes. Acinar cells, endothelial cells. So there's a lot happening in this disorder, in the classic version of this disorder, that we really need to start thinking more holistically about how cells interact with one another. And so, the thinking here, our lab has been focused primarily on beta cells and how they interact with immune cells. And beta cells release, we think, at some point in time, autoantigens that are recognized, carried by dendritic cells, presented to T cells. The T cells come in, and then they kind of mediate destruction. So that is an interplay that involves the beta cell. So there's nothing heretical about that statement. How do we kind of picture the disease? And this has sort of been the premise on which this project was originally conceived. And that is that, we think people start out with a pool of healthy beta cells. And then, at some point, there is some kind of an environmental insult. This environmental component is something that we have, I think, almost universally accepted in the field of type 1 diabetes, that, it's not genetics alone, it's not, inherent risk, but it's the combined, maybe genetic risk with some environmental insult. And that, we kind of believe that this environmental insult, let's call it inflammation in the most general sense, can then impinge on beta cells to cause stress pathways that are activated, such as oxidative and endoplasmic reticulum stress. That can then cause protein misfolding, and those misfolded proteins can become targets for post-translational modifications, which can then arguably serve as neoantigens that then get presented by dendritic cells to the immune system. And then you start to get this sort of cycle of propagation, if you will. And along that road, you might see things like, antigen spreading, and you start to get a variety of different autoantibodies and or a variety of different targets against which T cells are directed. So really, where does the process begin? I mean, does it begin with the immune system? Does it begin with the healthy beta cell? This is a question that we're asking. This is part of the field. But let's look at early disease and see if we can get some sense. So there's two papers that I wanted to kind of highlight that have been published, as you can see here, one in 2010, one in 2014. That looked at individuals, this PTD1 in the upper chart here is pre-type 1 diabetes. And then if you look down here in this paper, they were looking at individuals before autoantibody development. And if you look at those individuals, this early paper suggested that if they then did gene expression profiling using a microarray analysis from whole blood. That was way back in 2010. They found that some, clear subset of individuals have, interferon activated genes in whole blood. Similarly here, using, a screening array focused on the interferon signature, this group, Netta Ziegler and group here, identified that there was a statistically significant increase in an interferon signature that circulates in children that are genetically at risk for type 1 diabetes. And you could see that those who never seroconvert, don't show that same propensity to that signature. So the thinking here is that there's something circulating before the development of autoantibodies that could be an environmental component that could infect, we think, beta cells. So and keep that in mind, that interferon signature. We'll get back to that a little bit later. So looking at this pathogenic loop, I guess one question one can ask is, can we identify something that's circulating? I mean, it could just be interferons, but that alone, interferon signatures will increase in lots of people, as you could probably imagine, who might get any kind of a viral infection. So that alone probably wouldn't be a very specific biomarker. But we can ask, knowing what's happening in this disease, somewhere along the way here, can we identify biomarkers that can reflect the disease process itself? So what would those biomarkers be? Well, we were particularly interested in alternative splicing of mRNAs. And why is that? Well, there's a number of reasons. Alternative splicing is a mechanism that allows really multiple proteins to be generated from a single gene. And what that does is obviously increases the diversity of both the transcriptome and the proteome. So you don't have just one mRNA giving you one protein. You could have multiple mRNAs, giving you multiple other proteins. And alternative splicing has actually been proposed as a pathway that may unmask novel immunogenic protein epitopes. So that's important when you think about autoimmune disease, because aberrant patterns of alternative splicing have been studied in other autoimmune diseases or autoinflammatory diseases, such as multiple sclerosis, lupus, rheumatoid arthritis. And so, obviously, the question to ask is, what about type 1 diabetes? And recent studies in human islets that have been done, by our group, by, the group, in collaboration with Indiana, Decio Aziric's group in Brussels, have suggested that in human islets, splicing events may contribute to beta cell dysfunction and immunogenicity when they use an in vitro model. So, it's just throwing, interferons and pro-inflammatory cytokines on islets, and you see a pretty dramatic change. So, so those are all suggestive of a process that could be triggered or initiated by this sort of systemic inflammation, such as interferons. So our hypothesis going into this project, which was started some years ago and has actually been funded by and large by the Human Islet Research Network through a grant that I have been leading, that includes Pacific Northwest National Labs, Indiana University, and Brussels, that alternatively spliced mRNAs in the circulation may reflect disease pathophysiology. And we're not talking about, cause effect here. We're just talking about, associations, because associations may give us a clue as to, the propensity for developing type 1 diabetes. So what did we do? And this was our workflow. As you can see here, this was funded by the HERN, and it involved Carmela Evans-Molina at Indiana, who helped us collect samples from new-onset T1D subjects. Wenting Wu, who did the initial alternative splicing analysis, and Bobbie Zhou, who then subsequently did the machine learning. But what we did was we collected whole blood from individuals with new-onset type 1 diabetes. There were a total of 48 subjects in this entire study, and I'll tell you how they break down. But these new onset subjects are individuals who who presented with diabetes within, roughly 48 hours of diagnosis. We had two groups of people, and this is kind of important, and Bobbie Jo is going to emphasize the importance of this. We had healthy controls and new onset type 1 diabetic subjects, and we did this twice. So we got, 12 of each, or excuse me, yes, 12 healthy controls, 12 new onset. You could see in total that was 48 individuals, and you could see our gender breakdown. You could see the mean age, mean BMI, none of which were different between our cohorts. And then we collected whole blood, and then we deep sequenced this. And when I say deep sequence, we went about 150 to 160 million reads on what we call our sort of discovery set or test set. And then we had a validation set that had a slightly lower number of reads, about 120 million. And the reason why we went so deep was we wanted to identify very specifically the different forms of alternative splicing that occur. And so there are really 5 different types. There's a skipped exon, there's a retained intron, and then you could get alternative splice sites at either the 5' or 3' ends. And then you could get something called mutually exclusive exons, where you get one or the other that are mutually exclusive. So these are the different types that occur. And we went to look at total mRNAs, the different splice sites, how they distributed between healthy controls and those with type 1 diabetes. And then we compared that first set and asked, when we put it in a machine learning algorithm, how well does it hold up in an entirely independent second cohort. So I'm going to now turn this over to Bobbie Jo, who's going to tell us our findings.
So as Raghu mentioned, the first thing that we did was when team ran some clustering and statistics on the alternative splicing RNA and mRNA data. So what was pretty apparent right off the bat is that you could cluster. This is just pure clustering with no, input as to what the groups are, that the alternative splicing definitely separates out our controls from our new onset T1D patients. So that gave us some, good confidence that we're on the right track. Next slide. And the statistical analysis also showed that we were finding more things significant than you would expect by chance. Here's sort of the distribution of the different types of alternative splicing, which is really dominated by the skipped exon. You can see over 104,000 in our training set. Then you can see how it decreases if you start filtering and using an FDR threshold. I'm going to focus on this very low FDR of 0.001 because that is the threshold we use for machine learning. But what I want to point out here that sort of jumped out as quite interesting, and you'll see why this is interesting as we go through the presentation, is that the retained introns actually have 8% of the events detected are significant at an FDR of 0.01, right, where the expectation is no signal of 1%. So that's really great. All the other ones are also above what you would expect by chance. As we mentioned, we had a complete independent test set. It was even run at a different sequencing depth. So it is truly an independent validation set. And so what we're actually looking at in the test set is how many of those events that are significant in the training set are also detected in the test set. And so that really varies depending upon, so this is not how many are significant in the test set, it's what percentage overlap our training set. So again, the retained intron, 363 events in the test set overlapped with the 370 in the training set. For comparison, if you look at the mutually exclusive exons, you have 215 that pass that 0.001 FDR, but you can only find 94 of those in the test set. They're not necessarily significant in the test set, but they're there. So that's quite a dramatic difference. And then everything else is in the 80s, which is quite good as well for the other ones. Next slide. So we use a fairly standard machine learning protocol. So across the top is our training cohorts. We have the 24 samples. From that, we did our filtering, meaning we had to have a certain count threshold of 10 and an FDR of less than 0.01. So that gives us a total of P events. If we go back to that retained intron, that number would be 370. And then we take those, we put them into a random forest. We use cross-validation, and then we can get our results and see what the variability is, because random forest is a stochastic algorithm. With the validation cohort, as I mentioned, we want to base it completely on the training data. So when we talk about filtering the validation cohort, that FDR threshold is based on what we saw in the training. So what we're trying to get is, going back to retained intron, since I pointed out those numbers, the filtered data for the validation cohort would be that 363. So that's what we call P1. We saw 363 of the 370 in our validation cohort. That data moves forward. In order to run the machine learning algorithm, we do need those missing 7, for example. So what we do is we just compute those with the average so that they don't really have an effect on the machine learning algorithm. And then we run that through the random forest model generated on the training data, and then we can get our validation results. And in this case, we get variability because we have built a lot of these random forest models, so we can see how the validation data does on a lot of permutations of that random forest model. Next slide. So here's how we do. So the training set, this is based on cross-validation. So we can see actually the highest cross-validated is the 3' splicing. And then we have confidence intervals there of 95%. We put the retained intron at the top here in red, primarily because when you look over at the validation set, it is the one that ends up with the highest average area under the curve. So an area under the curve of 1, probably most people here know this, is perfect. 0.5 is random. So a lot of these cross-validated results are definitely overtrained. They do not hold up in our validation set. And this is really what makes this data set, I should say data sets, really valuable, as we can really evaluate that issue of just using significant markers in building and cross-validation of a model. Next slide. So this gives you a little bit more on the variability. Again, the retained intron, the blue and the pink sitting there next to each other shows that our validation data set or test data set and training really are much more aligned, and the other ones you see that big drop in the validation. And why is this? Goes back to what I pointed out on the statistics, was that if you looked at retained intron, you have much more overlap in those, not just the events detected, but the ones that are significant. So the blue and the yellow bars being much higher retained introns means that of those events that overlap between the two data sets, we had a lot more significant in both the test and train, and not just in the train, but not the test. So the results make a lot of sense also in terms of the statistics we saw. Next slide. One more thing we wanted to try was to determine if we combine different alternative splicing event types, could we actually get an even better accuracy? And we don't. So the retained intron alone does better than the retained intron plus the data from another splicing type. The second best being with the 5', but it is not statistically better than the retained introns on their own. Next slide. So we were fortunate, Raghu and I put together a supplement for to our Parent Hern grant to generate AI/ML ready data. So this was our flagship data set to sort of demonstrate the process. And this will all be released with the paper that we have about ready to submit. But really what AI/ML ready data means is just that it's cleaned and processed and it's in a usable format that somebody else can come in who may not be have in-depth knowledge of sequencing or diabetes, but there's enough information for them to put it into their own programming tools and evaluate their capability to do prediction or to reuse it for another purpose. And this bipartisanpolicy.org has a great description of AI ready data in that it should really have 4 core properties. So this is different than FAIR. FAIR sort of is really just about that it's findable and it's accessible and it's interoperable. It doesn't mean that you necessarily have quality or documentation and for reuse. AI/ML ready goes the next step. So by default, the data will be FAIR. AI/ML ready is FAIR plus something more, which is these 4 key components, which is the quality, that it's consistently formatted. There's documentation associated with the data and the domain. Its access is available in formats that many people can use it. It doesn't take very specialized knowledge to use it. And then it's gone through some sort of pre-processing steps to support the utility of AI/ML tools and software. So I think this data set's going to be of interest to a lot of people because we really have those 2 key pieces, an independent training and test sets validation data set. So next slide. So I'm going to show you a website here in a minute, but part of the process is generate what we call a model card. So it covers those core components. So the data, what is the data, what's available. So again, you can see some of the information we've already presented in terms of the cohort 1 and the cohort 2 are training and testing. How many events we have, shared events, what was our approach, just to give somebody else a, how has this been implemented to date? It's been implemented in R using tidy models and other packages. How did we tune the model? That sort of information, very high level, but enough for somebody who does AI/ML to be able to see how we have processed it to date. And then a piece that demonstrates the results, again, sort of these AUC machine learning results as well. In this case, we don't present that a lot in this presentation, but what were the key events that popped out of the model as being the most predictive? Next slide. So one thing I want to point out in terms of sort of data preparation and reuse, here's our GitHub page. It has all the different models that we use, and model tuning scripts are there and available for use. Next slide. And then we have a core page. So this is a publicly available page here on a tool we call Data Hub. And again, you'll see a lot of that same information. That model card, you can actually download it from here. Next slide. So as I mentioned, one of the key components of AI/ML ready is the documentation piece. So here we have an actual data guide. So it goes through every RDS file that you would get if you went and downloaded the data, as well as what each component means, so that another user could come in and understand each piece of the data. Next slide. And then the download where you can get to the actual data files. We have added, so these were all in R. We've also now added an Excel for somebody who wants to import it into a different tool rather than R. So that gets to the access piece. And next slide. And that takes us to the pathway analysis that Winting also did. And I'm going to let Raghu talk about the consequences of this analysis moving forward.
Yeah, so we did more traditional gene ontology pathway analysis using just the retained intron components that we found to be most predictive or correlated best with type 1 diabetes. And we did it from the two sets. So we had the training set, as you can see on the left side, and then we had the validation set or the test set that you could see on the right side. And I think that what we kind of got out of this is some things that maybe don't surprise you so much, for example, RNA splicing, spliceosome processing, and so forth that we did see. But one thing that did appear, highly significant in both was defense response to virus, and you could see that that shows up here as sort of the most significant hit, and this is one of the most significant hits on the right side. And this is again important. If you look at the genes, or at least the protein, the encoded proteins of the genes, they tend to all be in the interferon response pathway. So once again, now here, using alternative splicing, using a completely unbiased approach, we're able to find the same signature in very, very early disease that was identified using more biased approaches that I showed you earlier, using either microarray technology or very specifically PCR-based technologies. So it's for us at least a great validation that we're learning something about the biology. That what is interesting is this notion that retained introns seem to be a component of this biology, and many of these retained introns, as I said before, have been associated with other autoimmune diseases, or at least the differential expression of these retained introns, and that many of these may reflect loss of protein function. But at the same time could represent proteins that are neoantigenic in many ways, that could be disease causative. So we're not trying to make a link between cause and effect, but it does raise an interesting and intriguing possibility. And I want to just point out that we looked at individuals very early in after disease onset. So obvious next steps could be to go backwards in time, so to speak, and look at individuals who either have autoantibody positivity or before they're positive, and then that will raise additional interesting and potential biologic pathways that could be active. So our conclusions based on our whole blood RNA alternatively spliced analysis is that these retained intron splice variants appear to drive the best predictive capacity in random forest. That we didn't show you the data, but there is about a 7% overlap between what we see in the circulation and what we were to see if we were to look at primary islets. That's a previously published data set by our group. And the point being simply that systemic processes like you see in type 1 diabetes, will affect multiple tissue types, and that there may be shared signatures across these tissue types in the setting of inflammation. And that the other comment, and I don't know if you made that comment at all, Bobbie Jo, was that small subsets of these alternative spliced RNAs in the retained intron categories are unlikely to provide sufficient information. You actually need a lot of them. You need at least 150 in order to be able to say that you have an appropriate predictive capacity. So I think what it means is that there isn't any one species that's going to, or 2 or 10 or 15, that will give you predictive capacity with alternative splicing. And this may be true of a variety of different, whether you look at mRNAs or proteins, and that just tells you the complexity of the disease and the different cell types that may be involved in the disease. And where we could go from here is adding additional features that could, help improve our area under the curve in the receiver operator curve, such as adding proteomics and lipidomics, which we're doing as part of collaborative efforts between our group and PNNL. And so we're pretty excited about where this could lead down the road. Any comments to add to that, Bobbie, Joe?
No, that's great. Yeah, I did not mention, we did also evaluate by feature selection, and it required a lot of features to hit that top area under the curve value. So yeah, it's not just a few. It's quite complex.
Yeah.
Very complex model.
Yeah, it is quite complex, and you made it very transparent and understandable sort of across disciplines. So thank you both for that. Here's a question in the audience, Kevin Bodie. Great talk. I'm just wondering if you are using all mRNA reads to identify AS events control versus T1D, or if you can pre-select for known beta cell related mRNA to make it more specific.
So we're using all mRNA reads. So we didn't kind of drill down any further than that to look at beta cell specific, because we didn't actually go into this, thinking that we were going to identify beta cell specific signatures. They may exist, as you said, as I said, that there was a 7% overlap in the types of alternative splicing that we see. It's possible if we drilled down deeper, we might be able to find more, beta cell-like signatures. I would think that we would have to probably go earlier in the disease process where there's probably going to be more beta cells, and there's probably going to be clear evidence of either beta cell dysfunction or very, very early dysfunction. I just don't think our data set was really, built for, beta cells, so to speak.
Feel free, everyone, to drop questions in the chat or raise your hand.
I don't know, I know Wen-Ting is on the line, and I don't know if there's something she wants to add, because she's really been spending a lot of time looking at alternative splicing.
Wen-Ting, would you like to add something?
Yes, thank you, Raghu, for mentioning me here. I just think in the first you and the Bobbie had done very good work for those, and I saw there are lots of machine learning running, and they're exciting to see that RA has did a pretty good job. And I think it's very agreement with other immunology analysis right now into the alternate splicing that return intron really bring a lot of their immune antigen and for this particular category or alternate splicing. So that is consistent with our finding that return intron really did a pretty good job, even though they're only very minor percentage of the general alternate splicing events. So I feel that's very great work for the prediction in alternate splicing part. And based on your first slides introducing on the genetics part for the interferon gamma, I'm thinking maybe in future we can do more into those genetic risks, the family children for the prediction. If they have higher risks of the type 1D risks, we can narrow down to those risks of children further to see how their machine learning algorithm work. I'm not sure how you think about this. Just and a very nice talk today. Yes.
Yeah, I think, and I agree with Wen-Ting, going earlier in disease. And I think the other component, as we talk about interferons in general, I think it's really type 1 interferons. A lot of the alternative splicing that's been done in islets so far have been really the type 2 interferon, which is interferon gamma. But we're beginning to appreciate, the very early role of interferon alpha and potentially other type 1 interferons.
Thank you, Wen-Ting, for adding that value. I have a question for you. So when you talk about gaining your blood samples, what volume are you talking about? And, in terms of scaling and gaining more samples, right, particularly in this sort of pediatric, whatever, 5 and under or 12 and under population, what are some of the challenges you're going to have? And, you know, how might you kind of like scale this? So you could gain, you know, more, you know, this is like the typical question, how can we get more people involved, patients involved, or people in the prodrome involved? But how could you get, what sample is the minimum that you need? And how can you gain more of those samples so that you can run these more robust machine learning experiments?
Raghu, Raghu is working on clinical side. He might be more clear on that. I have a impression that it's not too much. Even when I do single cell RNA sequencing, that's not too much. Quite a few is okay. So Raghu has more idea because he's
Yeah, I mean, we're looking at really functional genomics here, and this is a little bit different because when you have to read sequences at great depth, you need, you know, since we're doing whole blood here, you need a decent amount of RNA, you know, not a drop, you know, and so yeah, it can be challenging. I don't recall how much we used, but I think it's definitely more than one ml for what we had to do in terms of getting this level of sequencing depth. So it's just something to keep in mind. Similar to what we, you know, see, for example, we've talked about it, maybe you've had sessions on this before, Monica, epigenetically modified DNA fragments, for example, require a fair amount in order to be able to sequence deeply. So I think that could pose a challenge, but you know, where we're at with this work is, you know, I think we're looking for a signature, and maybe somewhere along the way we can find that there will be, you know, only a few, you know, alternatively spliced RNAs that we need to interrogate in the context of proteins and metabolites and lipids, and maybe we can identify among all of those, if we can combine everything, a signature. But it's still at this point in the research, it's going to take a fair amount of blood.
Yeah, I think some notations would, like the pediatric research draws can't exceed 50 cc's.
Yeah.
So yeah, that's kind of interesting. Like, what are the limitations, or what are the, I guess, what are the workarounds? Because it does seem like this is a really interesting way to go. And I like the fact that you guys are really approaching it from a physiological standpoint, right? Like, what's happening throughout the physiology of a person as they move towards, you know, type 1, not just one side of the equation. Then I also wondered, you know, just sort of, this is sheer speculation, but if you think about the spliceosome, right, in context of interferon, would you say it's like overworked, you know, if it's not able to, like, you know, splice out the intron, or is it kind of like sluggish, you know, from some people have a sluggish spliceosome from the get-go? I mean, what about that piece?
Well, you know, just keep in mind that the retained intron component was one of the smallest, you know, changes that we saw in terms of absolute numbers. So there is actually a lot happening across all of the different alternatively spliced features. So you're seeing, you know, so I don't know if it's necessarily an overworked system. I think it's, you know, I think it's a highly active system that seems to be responding, and I would argue to, you know, perhaps different, you know, degrees of interferon circulating or, you know, different kinds of interferons, type 1 versus type 2. So I think this is something that changes dynamically with disease. I mean, this is different than genomics, right? This is functional genomics.
Right.
And so this is a dynamic process. And so the only way we're going to kind of get at sort of what's happening over time is to collect samples over time, preferably from the same individuals, right? Rather than, you know, different people. And we have that, right? I mean, those types of, you know, samples exist. You know, there's Teddy, and there's, you know, there's there were other networks and certainly the ones out in Europe. So those samples exist. It's just a question of, leveraging them for studies like this.
Yeah, access and then getting
Yeah.
all the studies done,
Yeah.
which takes funding. And then, so I guess my other question, and still welcoming other questions, regarding the interferon, you referenced alternative splicing implicated in SLE, RA, and MS. And I mean, are there already sort of like, known trajectories of what interferon does in those disease states that you can kind of cross compare to, or are they also kind of like just getting into the water with this kind of analyses?
They're a little farther along than we are, because, this type of work has been done, a few years before we initiated it in type 1.
Yeah.
But I think at the same time, you have to keep in mind, there was a nice paper by Decio's group that showed a lot of signatures that overlap between different autoimmune diseases. I mean, it was published in, I think, Science Advances a couple of years ago.
Yeah.
But there are still key differences between the autoimmune process that's happening in type 1 versus other diseases. I mean, for example, type 1 diabetes, shows, a completely different, demographic in terms of sex compared to other autoimmune diseases, which are going to be largely female predominant. In type 1 diabetes, the prevalence is close to 50-50, actually a little bit more male than female. And then if you look at when the disease begins, if it begins after puberty, it's actually almost 2-to-1 male compared to female. So it's a very different kind of disease. So, if we're going to go after, saying, hey, let's model what we see based on other autoimmune diseases, and have we learned enough from them that we can apply it to type 1? I think the rules are going to be different.
Yeah, I really was kind of getting at whether or not you could create any ML models together with these other disease progressions, but it sounds like no.
I think just given what we know of disease pathophysiology, I think it would be very tough.
Mm-hmm. Yeah. Well, this is fantastic. You just got another accolade, Scott Solomon-Poor. Congratulations on the impressive work, Raghu, Bobbie, Joe, and Wen-Ting. It was a great talk. I enjoyed and learned a lot. Thank you.
Thank you, Scott.
Yeah. So we're really looking, everyone is really looking, I think, after getting a taste of this work for the paper and what's coming next. It's really interesting. I love the fact that you are partnering with PNNL and, kind of going after some of this pre-existing data with this new approach. And it's really fantastic. You've got all the sort of powerhouses, IU, Chicago, and PNNL. And so congratulations to all of you.
Thank you.
Thank
Thanks.
you.